The shatterproof gene is a mutated gene known as SHP1 and SHP2. This gene is found in the nucleus as a transcription factor. These genes can cause the seed pods fail to shatter or burst. Due to it being a transcription factor, it happens when the mRNA is being created, and instead of it directly affecting the strand, it attaches itself on top of an already created protein. SHP1 and SHP2 are typically found together, which creates a double mutant. This double mutant makes the seed pods fail to disperse their seeds normally. The gene can either be too strong, resulting in a late seed dispersal, or too weak, resulting in the seeds bursting before they reach full maturity. In a late seed dispersal, not all the crops would be able to grow to their full potential, as the seeds have become old in the pod. In an early seed dispersal, not all the seeds would be ready to grow, therefore resulting in a low yield of soybeans. As discussed in the previous slide, the timing of the seed dispersal can have a great effect on the yield and ripening of the soybeans. No matter whether the seed dispersal is early or late, the soybean plants may not ever grow to be fully ripened. The overall results may be that the soybean plants are too short and there's very few of them. The double mutants also fail to allow the seed pods to split. The valves on the pod are not able to separate from the replum, which is the thin internal separating wall or partition between valves or compartments in some plants. Cell separation can't normally occur when the double mutant is overexpressed. So why should we care if the shatterproof gene is present in soybeans? Well, this gene is a major cause of yield loss in mechanical harvesting of soybeans, especially in tropical and subtropical regions annually. The soybean is a widely grown plant and is found in hundreds of foods. It is a good plant to be grown as it has a high nutrition value and it is not very hard to grow. That is, unless the SHP1 and SHP2 genes are present. To make this gene more efficient, there needs to be research done to find the correct place on the DNA strand where the shatterproof gene is expressed at just the right amount, not too little and not too much. In order to do this, there should be use of the CRISPR-Cas9, which is a unique technology that enables researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. It will allow for another mutation in the DNA sequence where it can modify the seeds so that they can shatter at the appropriate time to give them their full growing potential and yield.